next speaker is Jenny Lee. Jenny is a PhD student from UCL Queen Square and Miss Queen Square Institute of Neurology in London, working with Professor Seven Bestman and Professor Nick Watt. She's funded by Brain Research UK. Jenny is interested in how non-invasive brain stimulation techniques could be used as part of stroke rehabilitation. Please do find more information about her in the chat, and you also can find her on Twitter. I'll post it in the chat. Jenny, please take the stage.
Okay, thanks very much for that kind introduction, Gaiqing. As you said, I'm a PhD student in the ARM lab at UCL, and this talk is going to be about achieving opposite direction of tDCS current flow in neighbouring cortical sites, and why we might want to do this. So transcranial direct current stimulation, or tDCS, is a well-established non-invasive brain stimulation technique, and it can be used to alter cortical excitability. It involves passing a weak electrical current between electrodes placed on the scalp through the brain, as you can see on the left-hand side of the slide. And it has promise as a therapeutic tool in lots of clinical settings, including stroke rehab, because we think that altered cortical excitability could underpin the plasticity mechanisms that are important for relearning. So currently tDCS is more commonly seen in experimental settings in the clinic, unfortunately, because although it's very well tolerated, the excitability outcomes are often highly variable. In other words, it works for some people and not for others. So there's now a large body of literature out which is exploring how the variability in tDCS studies could be reduced. And often the focus of those papers is about the intensity and focality of the injected current. So ensuring that the same amount and the same amount of E field and the same area, the same cortical area is stimulated across all participants. However, direction is also important and it's often overlooked. So as you can see in the right-hand side of this slide, broadly speaking, inward current flow, which is traveling along a neuron towards the soma, is associated with excitation, whereas current flowing in the opposite direction is associated with maybe inhibition. So the point of this slide is to show that the direction at which tDCS current hits the area that you're interested in the brain is important, and we don't think about it very much at the moment with our current study design. So conventional tDCS, as you can see on the left-hand slide again, is normally applied with a posterior anode and an anterior cathode, so that current flows anteriorly straight forwards towards the front of the head. But due to the natural cortical folding structure of the cortex, the orientation of grey matter pyramidal neurons will alternate. So as you can imagine in this diagram on the right, if current is flowing forwards like this, and you have one side of a sulcus that's orientated like this, and then it moves around here, then you might get pyramidal neurons which are stimulated with outward current, which could be inhibitory. And then on the opposite bank of that sulcus, you could get inward current with the orientation of the neurons, which could be excitatory. So current flow in the same direction could actually have opposite polarizing effects for neighboring cortical sites, as you can see here. So we were interested in modeling a tDCS electrode montage, which might push current in opposite directions with respect to the whole head, as you can see here, which in fact is the same direction with respect to the neurons in these structures. So in this case, the target regions that I'm looking at are M1 and S1. This is because we're interested in motor rehabilitation after stroke, and most of our studies are to do with the primary motor and primary sensory cortices. These two target areas tend to be, well, they are found on either side of the central sulcus. So with M1 here anteriorly and S1 posteriorly here. And we want to try and push current forwards through M1 and backwards through S1 so that we might achieve the same polarization of the neurons in these two areas. And the main reason we want to do that is because the motor and sensory activity is so intrinsically linked that we think it's important to stimulate them in the same way. So to do this, we used ROSE current flow modeling software and prescribed anterior current flow through M1 and posterior current flow through S1. We then asked Rose to generate an individualized montage with up to 72 electrodes. And we used FreeSurfer to generate a gyri precise mesh of the grey matter so that we could look at what would be perfect inward current flow in each of these cortical areas and then compare it to the actual direction of electric field. And then we compared this suggested montage that Rose gave with a conventional montage that you can see here. Very briefly, what we found is that we can achieve posterior current flow in S1 and anterior current flow in M1 at the same time. But this came at the cost of intensity. So the blue areas here show that you get low intensity in M1 and S1. For example, about 0.02 volts per meter at the cortex, where normally with about 1 milliamp of standard current, you would expect an average of 0.185. And other studies that have just come out by Caulfield et al. Have shown that actually you might want 0.1 or 1 volt per meter in a target region to elicit neuronal spiking. So we didn't get enough current there, even though we achieved the direction that we wanted. So future directions are to generate a individualized custom montage with fewer electrodes that might still push current in the directions that we hope it to go in M1 and S1, but achieve a higher intensity, which is more comparable to other studies with injected current. And a physiological study to test the effects of this montage on excitability is ongoing, depending on COVID restrictions. So that's a lot of information. I'm happy to take questions. And thanks very much for having me.
Thanks very much, Jenny. That's a very beautiful talk. I'm really impressed by the fantastic and rigorous result and the beautiful figures. Anyone have questions? Let me check Q&A. We currently no question in Q&A. Let me see. Can I ask question? What's the next step for probably apply what you are doing to the rehabilitation?
Yeah, so with rehabilitation, we're running another study which is looking at whether tDCS, this brain stimulation technique, would be best applied immediately after somebody has a stroke in the next few weeks after that happens, or if it would be best applied later on. So we think that something like tDCS that could boost excitability might be useful because it can enhance plasticity mechanisms. And we're just trying to work out if that would be better to do early after a stroke when plasticity is already high, or if it would be better to apply later after stroke when plasticity levels or excitability levels have reduced back to kind of a normal state. So it's not ready to go into clinical settings yet, and we're still doing exploratory studies, observational studies with the clinical population. But we hope that in the end it might boost excitability to support the existing training that people get with physiotherapy and things like that.
Thanks very much. Very cool. We have a question from Alessio asking the montage is specific to the user. You can also click the chat or the Q&A to see the questions.
Oh, okay, I see. Thank you. With the protocol, so the question is, would the protocol differ as well with directly or after long stroke with regards to frequency?
Okay, we can answer this first, and we have a question in the chat. We can answer later.
Oh, I'm sorry. What
worries.
We need to answer?
As the montage is specific to the user.
Yes, so the montages are specific to the user. Apologies for that confusion. So with ROAST, what you can do is you input a scan. So the scans we used here, I forgot to say, were from the Human Connectome project. So this is an individual person's brain. It's not just a standard MRI scan. It's a 3D construction that's made by the ROAST software created by Hwang and the Paralab, which gives you a 3D model of the brain, places electrodes over it to achieve your desired direction and intensity of current, and then tells you which electrodes it suggests and what intensity of current to inject into those scalp electrodes. So it's a great bit of software. And yes, it does individualize the tDCS protocol for each person.
Okay, thanks very much. We have a question in the Q&A from Eva. I think you already saw that.
Yeah, I'm sorry about that. So before, so would the protocol differ as well after stroke with regards to frequency? So is this question, correct me if I'm wrong, if the protocol would differ depending on how long after stroke a person is? So the answer to that question is we don't know yet. So hopefully in future we'll be able to model with a bit more detail, taking into account lesions that appear after stroke. So for example, immediately after a stroke, a damaged area of the brain might be filled with blood or cerebrospinal fluid or other tissue, which conducts the current differently to cortical spinal fluid does, which is kind of what you see when you see that lesion about 6 months afterwards. So at the moment, we would keep the protocol the same, regardless of the time post-stroke, as long as it isn't too soon afterwards when the original trauma happened. But maybe in future, we will adapt our montages to take lesions into account in a more precise way, but we're not sure yet.
Cool, thanks very much. And Eva replied, yes, that's what I meant, thanks so much.
Thanks for your question.
Yeah, just tell you if anyone want to ask question on stage, just let me know and open your camera. Okay, cool. It seems no more questions coming out. I think almost time to move on to the next talk. Thanks very much again, Jenny, for your wonderful talk. Thanks very much for the questions. So, next speaker is Sututi